So now this is the lecture on the part 2, topic 3. Yeah. So for the topic 3, right, as you can see, the note is very short only. Okay. Because actually not much concept that you need to learn here. Okay. So of course, what is important, I will highlight it in the lecture later. All right. The major learning outcome or the major questions that can be asked for this part 2 lecture, right, is mainly on calculation. It's mainly on calculation. Okay, so in the class test 1, I think there will only involve one SAQ, and if not mistaken, that whole SAQ is about the calculation. So you have to ensure that you really understand, you really know how to do the calculation. Okay, if not mistaken, it's 5 over 30, so that will be around 20% of the weightage. Right, will be on calculation. Okay, then class test 2 only will ask about the part 1 lecture on the structure of the cell and organelle and all. Okay, but this is not finalized yet. This one depends on the module coordinator announcement. Okay, because module coordinator is the one that decides, right, the question. Okay, so that's why calculation, this one is very important. Then in the EOS, in the final exam, it will involve all. Then it will involve all the calculation plus the structure, cell and all that. Yes. Oh yeah, I think that should be the, because that file is actually under the OneDrive of Miss Abigail. So here she is actually. Yeah, so I think maybe she moved the folder or she actually renamed the folder or something. So that's why the link broken. So you all text Miss Abigail and then ask her to reshare the link. That should be fine. Okay. So then for the calculation part, it will also involve for the practical also. The practical 2 also. The questions of practical 2, right? It will be involving calculation as well. Okay. So that's why no matter what, ensure that you are prepared for that. Okay. Don't. Well, I would say based on my experience, normally class test 1 biology is the culture shock that a lot of students will have. A lot of students will fail the class test 1. Yes. Okay. So reason is not that it's very difficult. No. The reason is because they don't take it seriously. Because the class test 1 for bio 1, right, is normally after math and English. Then math and English, I don't know, maybe it's simple. So a lot of the students thought that class test only. Class test. So when they go through the math class test 1, the English class test 1, they will feel like it's easy. But then when they come to bio 1, they have culture shock. Okay? So that's why make sure that you are well prepared for that. Don't play with that. Don't play with class test 1 because it's 50 marks. Okay? It's 3 grades in your final grade. Yeah? So that's why don't play with the class test 1. Okay? Make sure you are well prepared for that. As I mentioned, for this should be for topic 3, the SAQ should be mainly all calculation. Okay, so that's why make sure you are prepared for that. And if let's say you are weak in calculation, don't worry. I have series of the ways to actually help you to master it and score. That's the most, because our syllabus is actually very simple. Okay, it's just that whether you want to put in effort or not. Okay, so I have summarized everything, and then if you don't know, you are not confident with your weak in your calculation for this, you just follow my step. If let's say you are very confident, then it's fine. Okay, that's fine. You want to use your own way, it's fine. Okay, so this one we'll discuss it during the lecture later and also during the tutorial. Okay, so now we move on to the learning outcome. Yeah, so I hope that the 2 groups that I teach, no one fail. The other group I don't care. Okay, but then these 2 groups, please. Okay, so hopefully I can proudly say no one fail in my class. Okay, so please put in your effort. Okay, be prepared for that. Okay. All right. So for learning outcome, same thing. We will look at the mind map. Okay. So this is the learning outcome. All right. The first part we will look at is okay. First thing first, you can see the first 3 parts is more into theory and concept. Theory and concept actually not much can be asked, and I can tell you it won't be asked in the SAQ because learning outcome is not
it is not in the SAQ. The learning outcome for this whole part is actually in MCQ. Okay, so SAQ is mainly confirmed calculation. Alright, so that's why when you prepare for the exam, when you do your revision, yeah, you know what is the strategy that you need to use. Okay, so now throughout the lecture note, I'm going to highlight all those concepts, all those important concept, and also the theory that you must memorize. That's it only. The rest, extra one, no need. You don't have to memorize word by word. It's not that important. Okay, so now first part is the microscope, type of microscope. Okay, so for the type of microscope, we will learn two categories: the light microscope and also electron microscope. Okay, so the light microscope one we will learn for 1, 2, 3, 4. Yeah, so the bright field, dark field, phase contrast, and fluorescent. While for the electron microscope, we will learn 2, transmission and also scanning electron microscope. So total 6 types of microscope. Okay, so I think you look at the note, there is actually quite many descriptions, right? But what is important? Yeah, what is important? What will normally be asked is normally is only in one MCQ. Okay, they will normally ask, well, first thing is image. How the image looks like. Based on the image, do you know what type of microscope is that? Okay. Second, what is the application? Means that when you want to use it, why you want to use it. Okay. When you want to use the dark field microscope, why you want to use the dark field microscope. Okay. Phase contrast. Why you want to do this? Why you want to use it? That's its application. Okay. Straightforward. They will ask like this: why you want to use it? When you are going to use it? Or choose the best microscope to be used. Something like that. You have a situation. You have the story. Choose the best microscope to be used. So this is the only thing that we have. Okay, so that's why you need to know where you want to use it. Okay. Then besides that, right? Sometimes they will ask for advantage or disadvantage. What is the advantage of using the dark field, or what is the advantage or disadvantage of using the light microscope versus the electron microscope? Something like that. Okay, right? So that's why you can see versus. Sometimes they will ask like that. Okay. So then besides that, you have to know the resolution and the magnification for this microscope. Okay, it is listed in the table. Yeah, that number is important. Light microscope, all of them, their resolution is 200 nanometer. Okay, their magnification, 1000 times magnification, that's for sure. Electron microscope, TEM, and the SEM, different resolution. One is 2.5 nanometer, another one is 20 nanometer. All of this number you need to know. Okay, because this is those straightforward questions can ask. All right, and that's it basically. Yeah, the type of the microscope. Okay, then next we look at the part of microscope. Part of microscope, this one is mainly for the light microscope, for the bright field microscope. For this part of the microscope, right? I think you can see in the note there's a picture and so many parts. Actually, this part you have learned that before. Okay, how to use it? This one, well, we will talk about this during the practical because there's no point that I'm talking about it now. You have nothing to see, but nothing to show. Okay, so it's a waste of time. So that one we will review during the practical session. Okay, so but we will learn about the concept here, the objective lens. When you are going to use the objective lens, because there are 4 types of objective lens. Which one you should choose? Yeah, which one you should choose? And this concept is important because in the practical you have to choose the objective lens. We will ask you to draw, but then you have to decide what is the magnification that you want to choose. When you are when you are observing the plant cell, onion cell, when you are observing the chick cell, what is the best magnification that you should use? Okay, you have to decide yourself. Okay, so the concept, yeah, later we will talk about this. Okay, and then over here you can see the line. Yeah, cos node, we will only use it to adjust 4x and 10x. When you use 4x or 10x, you should only use the cos node to adjust it. And if let's say you are using 40x and 100x, you should only use the phi node to adjust it. This is very important because this is about money. Okay, we will talk about this during practical. This is about money. Okay, why? Because if you use the wrong node, it will break, it will scratch the lens, and then it will mean catching. Okay, yes, touching into IMU pocket, not me. Yeah. Okay, so please remember that. Yeah, so this is something that, yeah. But today we are more onto when and why we want to use it. Okay. All right, then next principle. For principle, you only need to know the definition. You only need to know the definition and then relationship between the resolution and also the wavelength. That's it. Okay. That this one very small part only. Then lastly is the calculation. For the calculation, yeah, question for sure it will be twisted. You have done the question for maths and chemistry. Calculation, first 5 topics all calculation for chemistry. Okay? Well, the question will be twisted here and there. It for sure will be twisted. Okay? Anything related to calculation will be twisted here and there. Okay? But no matter how they twist, right? Our questions is normally 5 types only. Okay? 2 categories, 5 types. Okay? So either it is the questions that is applying the magnification formula, or it is a question that using the stage micrometer to measure the sound. Either one category only. Okay? So under the magnification formula, there are 3 types of questions only. Straightforward, image given, scalar given. Only 3 types. No matter how the question twists, this is the only 3 types. Okay? And the major problem that the student have is they confuse. The question asking you to measure the cell, but they apply the magnification formula. The question asks you to apply magnification formula, but you use this way to solve it. That's the major confusion. So that's why the first step when you see the question is you try to categorize it. This question belongs to which one? Okay? If the question belongs to magnification formula, then only then you further see this belongs to which one. Okay? And then green color one is actually the first step to solve it. Okay, how to solve it? The first step, how to start the calculation. Okay, because the major problem over years, the major problem that the students say they have is they don't know what to start. They don't know how to start the first step. Okay, so this green color one is basically the first step to start. Okay, and then this one is actually the 3 steps to solve the question. No matter how they twist, this is the 3 steps that can solve every single question. Okay, it's just that you have to find the correct category only. Okay, so as for now, maybe it means nothing. You don't understand what it means. Okay, so it's okay. Later on, yeah, later on in the lecture note, we'll see one by one. So for measuring cell, there are 2 types also: estimation via the diameter and also measuring using the eyepiece reticule. Okay, so to categorize this one, very simple. If the question gives you only stage micrometer, it is under this one. If the question gives you eyepiece reticule plus the stage micrometer, it must be under this one. Okay, so once you can find once you can know which category is that, then you just follow the 3 steps that. Okay, so it's actually very simple. I have summarized it to simplify it, just to become 1, 2, 3 steps. That's it. Okay, so that's why if let's say your logical calculation is weaker, then you can always use my step. It's fine. Okay, but if let's say you have your own way, some people they want to use their algebra way to solve it, some people they want to use ratio way to solve it. All of this will be accepted. It's just that in the marking scheme, you have to ensure you can score. You have to write certain steps in order to score. This one we'll discuss during the tutorial. Okay, regarding the marking scheme. Okay, but if let's say you follow my way, it's confirmed following the marking scheme. Okay, because this is a general way to solve everything. All right, so with that, that would be all about the topic, the part 2 lecture here. Okay, so we will start with the type of the microscope. So the very first part, introduction. Robert Hooke, I think this one everyone knows him. Okay, Robert Hooke is the first one that used his microscope. The first one to use the microscope. Okay. So the first one to use the microscope to observe the cell and name it as a cell. Okay, so Robert Hooke. This one not important. That's it. Okay. Then next, after a few years, right? Then Anthony van Leeuwenhoek, right? So he actually produced a second microscope. Okay. So this microscope is very powerful. Although it looks like very simple and then very small. Okay. Because you will put your sample here. you will put the sample here. Okay, just here. Just very tiny sample you put here. Then you put here. And then you see the lens very small also. So the size is around like 1 thumb. Okay, so you will hold it like this. You will observe like this. Okay, even though it's so small, so simple, but then it's very powerful. Okay, it can, during that time, this is like a few hundred years ago, they can observe 300, up to 300 times magnification. That's a lot. Okay, so that is the Anthony Van Leeuwenhoek. Okay, not important also, won't be asked. Confirm. We won't ask that stupid question now. Who is the first one to observe the sound? Right? That is for primary school maybe, okay? Not for foundation study. Okay, so now next type of the microscope. Okay, that's just a brief introduction. Okay, so light microscope. So this is the light microscope is basically those microscopes use the source of illumination will be the light. Right? It will be the light source. Okay, then they will use the two lens system to do the magnification. That is for sure, right? For light microscope. Okay. So the light microscope, the most common one, it will be the bright field microscope. Actually, in the market, there is lots more. It's just that for our viewers, we only look at 4 types that is more common one. Okay. So the first one is the bright field. Bright field is exactly the one that is common one that you use in secondary school. You use in later on in the practical. You are going to use it. Okay. One day when you work in a lab, you are still going to use it. You want to do master, PhD, you will still use it. That is the so-called compound microscope, bright field microscope. Okay, the most common one. All right, that means that without doing any modification. Okay, so for this one, right, first thing, you look at the image first. For the image, the background will be white. Background should be white color. The specimen, the cell, okay, it will appear colorless or colored. Colorless or colored. Okay. Most of the cell, most of the animal cell, except the blood cell, red blood cell. Okay. The rest of the cell is all colorless, almost transparent. Okay. So you can see it under the bright field. It's just that maybe not that clear to be seen. Maybe you can see the nucleus, but then you cannot really differentiate the vacuole, all this. Maybe you cannot see that. Okay. Plant cell, yes, plant cell by nature they are green in color. Okay. For example, leaf cell, they are green in color because they contain the chlorophyll. But some of the plant cells, like onion cell, they are colorless. Okay. So those that's colorless one, it's very hard for you to observe it. Okay. So in order to increase the contrast so that you can observe it clearly, normally you have to do staining. Staining, S-T-A-I-N. Staining means that you color the specimen. So this one is stained. And you can see this is the parameter that's being stained. Okay. So for bright field microscope, it's basically for general use. It's used for everything. Okay, that's when you use it. Used for everything. If you want to observe wet mount means you want to see the cell happily living inside. Okay, so you can see the cell moving. That one you use bright field. Okay, if you want to see the cell that sticks and then stained with different color, and that one you also can use the bright field. Okay, that means that under normal circumstances, under normal condition, you can always use the bright field option. Okay. So as I mentioned just now, light microscope, right? They use how they magnify. They use two lens system. The two lens system, one lens system is called ocular lens. Ocular lens. Okay, ocular lens is always 10 times magnification. Always 10 times magnification. Okay, and then objective lens. Objective lens, it can be 4, 10, 40, 100. You can choose one. Okay, you can choose. Okay, so to calculate the total magnification is always taking the power of the objective lens times with the ocular lens. So let's say you are using 4x objective lens. So the total magnification will be 4 times 10 to the 40. In practical, don't write wrongly. Yeah. A lot of the students, they see, oh, okay, I'm using 40x objective lens. So in the drawing, the title of the drawing, they write total magnification is only 40x. That's wrong. Okay, so please remember, total magnification, remember to times with the ocular lens. Okay. So done, then next, we look at dark field. Dark field microscope. Dark field microscope, their image, you see that their background will be black. Background will be black. Okay? The specimen can be color, can be colorless. Either one, can be color, can be colorless. Okay? So background will be black, specimen will be bright. Okay? So when we are going to use it, why you want to use it? Okay? Dark field microscope is normally when you cannot see clearly, even you stain it you stain the specimen. Under the bright field microscope, it shows that structure. You stain it already, the structure is still blurred. You are not able to confirm that. Then you can use the dark field to confirm. For example, you see this picture. Over here, you see that it's like something a bit blur, like blue. Okay, it's actually a cilia. But then we can't confirm it. It's blurred. Yeah, it's very blurred here. It might be the residue of the staining. We are not sure. Okay. So based on this, how to confirm that? Then we use dark field. With dark field, you can see it is a full round of the blue color. Full round of blue color, that means that this is a cilia. Right? You know that it's full with the cilia. It's not just one side. It's not just one side. It's like the whole cell having the cilia. That's how it works for dark field. Why you want to use it? When you are going to use it. Okay. Then that means that dark field can increase the contrast. Yeah, they can increase the contrast because their background is black. Okay. Then next, phase contrast microscope. Okay. Phase contrast microscope actually is quite similar. The usage is quite similar to dark field. You see the image. Image is bright. Okay, the background is bright. Okay, background is white in color also. Okay, it's just that the specimen, the color of the specimen will be darker compared to the bright field. Right, the specimen will be darker a bit. So that will mean that it will increase the contrast. It will make this even clearer. In fact, phase contrast, you can see the cilia. You can even see one by one the cilia. You can see the detailed structure of it one by one. Not just a line like this. And not just a layer like this. You can see one by one. All the cilia, one by one. So it gives you even more detailed structure. Okay. So when we are going to use it, okay, same thing. If let's say this one don't give you a clear image. They don't give you a very clear image, you want to further confirm, yeah, you can use this one to confirm. Okay? Or if not, this is very good if let's say the specimen cannot be stained. If the specimen cannot be stained, cannot be fixed, then phase contrast is the best actually. Okay? Fixed means that you, for example, you have a slide, right? Okay? If let's say you just drop one drop of the dirt on it, okay, and then you want to see, let's say you just drop on it, or you didn't dry it. You didn't stain it, you didn't dry it, you just put on it, and we call it wet mount. You just put one drop, then you put down the color sheet, that is wet mount. Wet mount, the cell is not fixed, they are still moving, able to move. They can be alive. Okay? But then, if let's say you want to stain it, right, you have to fix it. That means that after you put the sample onto it, you have to use the frame to dry it. So this fixed onto that. When it's fixed onto the slide, only you stain it. Okay, so you fix means the cell is dead. Okay, but for some of the specimen, you want to see the movement of the cell. You don't want the cell to be dead. And then you cannot stain the cell. If you stain the cell will be dead also. Okay, so that's why in that case, how? If you cannot stain the cell, you cannot fix the cell, and then it's still not clear under the bright field, then what else can help you? Phase contrast microscope can help you. Okay, phase contrast. So that's when you will use it, phase contrast. But of course, phase contrast microscope is expensive. Yeah, it's more expensive. So one day when you are doing your master project or PhD project, it totally depends on the supervisor. If the supervisor is rich, just getting a grant from the government, then yeah, you can buy a phase contrast microscope. Okay, it's just an addition of the diffraction plate. Okay, but that small diffraction plate will cost you like a few thousands. Okay, but it will give you a very good image. So if let's say your supervisor is broke,
Then the alternative is you choose dark field. Dark field is cheap. 50 ringgit you can settle it. It's very cheap only. You just buy the frame only, very simple. Even you don't have 50 ringgit also never mind. You use the 50 cents, you can create your own dark field also. It's just a modification of the bright field. Okay, alright. So this tree is almost the same one. It's just that the image produced, which one is clearer, this contrast is clearer. Later I will show you more example of the image. Okay. Then lastly is fluorescent microscope. Fluorescent microscope is special, different. Okay. This one you don't use the normal light. We don't use the normal light bulb. This one you will use the UV light. Why? Because we want to make the specimen grow in the dark. We will use the fluorescent technology so that it is staying with the fluorescent and then it will grow in the dark. Okay? So that's why you can see the sample. The background is totally black, dark, okay? And the specimen will grow. Okay? It's not just color only, it will grow in the dark. Okay? So when we are going to use this is important if you want to detect the pathogen or you want to detect the protein. That means that you want to detect the cell that will produce certain protein, or the cell or the pathogen is mixed, or covered by other things. That one it will help them detect the pathogen, detect the protein. Example, detect the protein. How? For example, have you ever heard about cancer marker? Cancer marker. Yes, right? Cancer marker. That means that if you go and do the full body checkup, it will involve the cancer marker. If you do the simple one, then it won't involve that. Okay? So cancer marker is a protein that will be produced by the cancer cell. And some of the cancer marker is that the cell supposed to be producing that protein, but then due to cancer, the cell stop producing that protein. Okay? So under the microscope, can you differentiate which one is cancer cell, which one is non-cancer cell? You don't know. Okay, can you tell you got cancer or not? You don't know. Okay, so bright field, dark field based contrast, you can only see cell. You can't differentiate it. Okay, but the protein will be produced. So some of the protein, you can make the protein fluorescent, make the protein grow in the dark. So with that, under the fluorescent microscope, you can clearly see which are the cells that will produce that protein. You will know that, these are actually the cancer cells. So if let's say all these cells with the cancer marker protein, there are a lot. And if let's say the whole tissue is invading into some other part, then you know that it's malignant. If let's say you see the cell is all these cells with the blue dot with the cancer marker, they are just quarantined at that one side, they don't invade, they don't spread, that means that has been done. That's how it works. Right? So with fluorescent microscope, you can easily do that. You don't need to break the cell, then do the DNA extraction, then do the PCR, then do this testing and that testing. That will be too time consuming and too cost consuming as well. Okay, but using the fluorescent microscope, you can easily solve this. This is one of the examples why you want to detect the protein. Okay, so you can see a lot of examples here. So this one, bright field microscope, you see red blood cell and white blood cell. This is before staining. Can you differentiate which one is white blood, which one is red blood cell? You can't. Okay, but after staining, yes, you can even know. Okay, this one is actually the what we call the lymphocyte. And this one is actually neutrophil. You can even know what type of white blood cell is that. So staining will increase the contrast. Yes, staining will increase the contrast. So that's why in bright field microscope, staining is very important, because without staining, most of the time you can't actually see any value in the image that's being produced. Okay, then next you see dark field. This one is the bright field. Bright field without staining. Bright field without staining, you can't see anything. You don't know which one is white blood cell, which one is red blood cell. But then for dark field without staining, you can know that. Dark field without staining, you know that all of these are the red blood cells. And then this is the neutrophil. You can see that neutrophil. It's not that clear,
not that clear, but then at least it can allow you to differentiate it. Okay? Thus, if let's say you are rich, you can use the phase contrast. Okay, this one dark field cheap. Okay, so that's why it's a bit, the quality wise is not that clear. Okay. Then next, phase contrast. This one. Phase contrast, you see, even sometimes under bright field, you see that you stain it already. The image is not clear. For example, in the study of the hematology, you need to study the shape of the red blood cell. You will learn it in the bioscience. Okay, you see the shape of the red blood cell, you know what disease you have, right? So, if let's say it's like blur and pale like this in the bright field, even the staining is still pale like this, then how? Okay, then you can use phase contrast. You will increase the contrast. Phase contrast increase the contrast, and you can see the shape clearly. Or even you see the cell under the bright field, it is blurred. You don't know where is the nucleus also. But then after the staining, you can even see the vacuole. Okay, so that's for phase contrast. All right. Then the fluorescent microscope, for example, this one. This one is for detect the pathogen. You have to detect the pathogen. For example, you see pathogen can be detected easily now. Why you want to use the fluorescent? Not for fancy problem, yeah. Okay. Example, yeah. Okay. Those patients with the lung infection. It has a lung infection, so what you need to do, the doctors need to detect what is the bacteria. Then only we can decide what is the antibiotic we want to give. Okay? So you draw out the sputum. When you take the sputum in the lab, you have to put under the slide, and then you have to stain it. Okay? So you put under the slide, okay, and then you prepare the slide with the sputum. So sputum is rich in mucus. Okay? And all those many rubbish. So how it will look like under the microscope? After you prepare the slide, nicely prepared, and then stain it, this is how it will look like. Can you see any bacteria inside? You see nothing. Because sputum is so disgusting and dirty. It's a dirty specimen. You will block everything. There is bacteria inside. It's just that you can't see it. It's blocked by all the rubbish. Okay? So how are you going to see that? Because the first step in the lab, yeah, in order to identify what is the species of the bacteria is that you have to know what is the shape of the bacteria. This one you can't see anything. How? Fluorescent microscope can help you. So the same thing, okay? So under the fluorescent microscope, the bacteria will grow in the dark. So with that, can you see what is the shape of bacteria? Very easy. This is bacillus. All of these are bacillus. You can see that bacillus, rod shape. Bacillus. So from there we do the biochemical test. Then only then we can know the species, and then when we know the species, we can test on that. That's how it works. Okay, so luckily we have the fluorescent microscope. You can detect the pathogen easily. Okay, all right. So that is the 4 types of the light microscope. Yeah. Then next we compare the light microscope with the electron microscope. Light microscope and electron microscope, right? Okay. They are different. Yeah, they are different. They use totally different theory to function, do different mechanism to function. Okay, so from here, what we can compare is that first thing, the source. Light microscope, we are using light. We are using visible light or UV light as the light source. Okay. Well, for the electron microscope, we are using electron beam. We are using electron beam. Yeah? It's a beam of electron. Okay, so that's the first difference. Second difference is the lens. Okay, so the light microscope, no matter what type of light microscope, they are using 2 lens system, and the lens is glass lens. 2 lens system. So that will be the eyepiece lens, or the also called ocular lens, plus the objective lens in order to do the magnification. Okay. Well, for electron microscope, they are not using the over here I see a condenser lens. Condenser lens is not involved in the magnification. Condenser lens is not involved in the magnification. It's only the objective lens and the ocular lens for the light microscope. Well, for the electron microscope, their lens system is not the glass lens. Yeah, they are using electromagnetic lens. That is the second difference. Electromagnetic lens is different lens system because they are using electron beam. Okay. Then thirdly, the image that's being produced. Okay. Image that's being produced by the light microscope will have color. They will have their natural color or the stained color. Okay. While for the electron microscope, no matter it's the transmission or the scanning, the image is always monochrome. Means that there's no other color, only black and white. Only black and white only. Okay. So that's the three differences. Okay, so for electron microscope, first thing first, the electron microscope is so powerful. Yeah, compared to the light microscope. Yeah, compared to the light microscope, they are so powerful. Why? Because they have way higher magnification, up to 100,000 times. Light microscope maximum 1,000 only, but the electron microscope is up to 100,000. Actually, with the advancement of the technology now, the magnification is even higher now. It's just that in our syllabus we don't follow that. Okay, our syllabus is still follow that, but if you search in the internet, the electron microscope now, they can even observe the atom. Yes, they can. Atom, yes. It's up to 0.1 nanometer there is resolution. That means that you can technically see the I think electrons still cannot, but the proton and neutron you can see it. Right? So it's very powerful now. The electron microscope is super duper powerful. It's just that our syllabus is still stuck at 10 years ago. It's still 100,000 magnification. Okay, our syllabus, if the question asks you, it's still 100,000 magnification. Okay, resolution wise, it's very powerful also because of resolution. The light microscope maximum resolution 200 nanometer, meaning that whatever that's smaller than 200 nanometer, you cannot see it. That means that that's why a lot of the organ now we cannot see it under the light microscope. Rough ER, smooth ER, Golgi apparatus, vesicle, all of this you cannot see. Okay, because it's smaller than 200 nanometer. That's our resolving power of this resolution of the light microscope. Okay, but for electron microscope, TEM is the most powerful one because it's 2.5 nanometer. Scanning electron microscope, 20 nanometer. So that's why it's more powerful than the light microscope. They can see more detailed structure. Okay, so that's why electron microscope, right? They are used to see the ultra structure. Ultra structure will means that all those structure that's so tiny, like virus, so tiny, light microscope compound cannot see, but electron microscope they can easily see that. Also, they want to see the internal structure also can. Internal structure, that will be, for example, inside the cell of mitochondria. And you can even see what is inside the mitochondria. You can see the crystal. So that's the penetration power. Okay? So that is for electron microscope. So they are using electron, not light. So that's why the process of the specimen, it will be very troublesome. And it will need very skillful, very skillful practice that only then you can process that. Okay? Why? Because they are using electron beam. So that's why the specimen needs to stay or actually coated, not stay, coated with the gold or platinum. Gold or platinum. Okay? So, well, if you coat the bee or the spider, I didn't say easy because at least you can see it. But imagine you need to coat the cell. How to coat the cell? So that's why at the very beginning you are going to spoil a lot of specimens. So you have to train yourself. You have to really train yourself, that only you know how to use it. Okay, it takes months to train. Okay. So there are two, There are TEM and SEM. Okay. So for TEM, for the TEM, first thing first, we look at the image. TEM, the image produced is 2D internal structure. It has high penetrating power. That means that you can even see what is inside the cell. Okay, but it's 2D. It's 2D. Okay. Well, for the SEM scanning electron microscope, it's 3D. But it is surface image only. That means that you cannot see what is inside the cell. You can only see the surface image. Okay, but you can see a 3D image like this. Okay. Then second thing, the electron pass. Yeah. So this one it will pass through the specimen. This one it will be just scanning the surface. So that's why this one is surface image. This one is in the internal structure. All right. Then how to process this? Well, for this one, it needs to be ultra thin section. Okay, that you put with the metal salt, right? Heavy metal salt. Yeah. But for SEM, it is basically coated with the gold or platinum. All right. Then this magnification and resolution, this number is important. This number you must memorize it. Magnification and resolution, you must memorize it. Right? It's very common. Okay. Then next we will see, all the image. Okay. So for TEM, right? This one is for light microscope where you compare. Light microscope, you see the white blood cell, red blood cell, red blood cell, white blood cell. Okay. So now we see how the TEM will be. TEM, they can enlarge. You can even see the capillary. This is one capillary. So capillary is only allow the blood cell to pass through at one time. Yeah, see this is capillary. This is blood cell. Okay. So it's having high penetrating power. So maybe looking at the red blood cell, it won't help you to see how high their resolving power is. Okay. So now we change to virus. All of these are herpes simplex virus. Right? Herpes simplex virus that causes herpes, the STD. Okay? So for this one, virus under light microscope, you cannot see at all. You can only see it with the electron microscope. Okay? Even at 1,000 times magnification, you might say it's due to the magnification. No, it's not due to magnification power. It's due to resolving power, due to the resolution. That means that at 1,000 magnification, right? Okay, light microscope see nothing. But under electron microscope, even at 1,000 magnification or so, you can see the dot there. Then only you further zoom in. So how come one can see the dot, one cannot see the dot? Even it's still there. The same slide also, one will see the dot, light microscope will not see the dot at all. You see nothing. Why? Because resolving power, resolution. That's the key. It's not due to magnification, it's due to resolution. So when you see the dot, then you further zoom in, then only you can see that. But of course, light microscope cannot further zoom in. They cannot even see the dot. So that means light microscope, their resolving power, their resolution is low, magnification is also low. Okay, so they cannot see the ultra structure like the virus. This one, another virus, you can further zoom in, not just looking at the virus, you can further zoom in the virus and see what is inside the virus. So this virus, very famous virus, anyone know this? I think 5 to 6 years ago. No? It's those. Huh? No? H1N1 is that one is corona. Corona is in overship. This one very famous 5 to 6 years ago. Ebola, yes. Ebola virus. It caused many deaths in Africa. Okay? And then our whole hospital system is like so alert with that, so nervous with this Ebola virus because you just scared that it will spread to Malaysia in that time. Right? Then this one you can see now, bacteriophage. This is a virus also. You see virus are landing on the bacteria cell, on the E. coli. You can even see that this bacteriophage is actually injecting the DNA. You can see that. Why? Because TEM is having high penetrating power. You can see what is inside the cell. Okay? You can even see the DNA, injecting the DNA. Alright? And then some of you might say, so how come this one is color? Well, this one is due to the advance in the computer science. They add color. The computer adds the color itself. But the actual image that's being produced is actually monochrome, black and white. Okay, but the computer software add the color. Okay. So done with TEM, now we look at SEM. Same thing, blood cell. Okay, so this is red blood cell inside, white blood cell. You cannot see the nucleus inside because this is SEM. SEM is only for surface image only, no penetrating power. Okay, so same thing again. The initial image should be black and white. It should be black and white. Okay. This is the herpes simplex virus. That's not. Alright. You don't see the nucleus because no penetrating power. This one Ebola. Ebola virus, the blue color one, every one line is one Ebola virus. So you can see this cell, the yellow color cell, is being attacked by how many Ebola virus? This one common line. Okay. Now this is the bacteriophage. You see they land on the cell. Okay. But you don't see what? You don't see they injecting the DNA. You can't see that because no penetrating power. SEF is only for surface. Okay. So besides the virus, besides the ultrastructure, you can also use it to see the bigger structure. For example, the free. You can see that also. Okay. So these are the usage for this TEM and also the SEF. Okay. All right. Then next we will look at the part of the microscope. Yeah. Okay, for this one, right? Okay, this is just a part of microscope and what is their function. Okay, actually most of it you have learned it before. Okay, ocular lens. What is important here is ocular lens, objective lens. This is the most important one. Okay, and then next is the stage. This is where you put the specimen. Also very simple. Okay, coarse node, fine node. This is where you move the stage. Okay, coarse node will move the stage a lot. Fine node will move it a bit. Okay, how to use it, when to use it, this one we will discuss it during the practical. Okay, so what is extra thing? Right, what is the extra thing that not that maybe you didn't learn before is that the condenser and also the diaphragm. Because there's a condenser lens here, just under the stage. Yeah? Condenser lens is a lens, but it's not involved in the magnification. Not involved in magnification. Yeah? Condenser lens is just to focus the light onto the specimen. So the light come already, then it will focus onto the specimen. That's a condenser lens. Okay. Then another one is the diaphragm. Diaphragm is the adjustment here. Diaphragm is actually to control the amount of the light to go into the condenser lens and go into the specimen. That's the only two things that's new to you. Okay. So with that, that's a part of microscope. That's it only. What we will focus on today is the lens, the objective lens. Objective lens, there are 4 types in general. Okay, they have the universal color code here. Okay, there are 4 types universal color code. Red color will mean 4x. Blue color will mean 40x. Yellow color will mean 10x. White or black color will mean 100x. Okay, so there are 4 types. Okay, so for 4x, for 4x objective lens, it is also called scanning objective lens. Scanning objective lens. Okay, why you want to use it? You want to use it to scan the gross structure of the tissue. We want to scan the gross structure of the tissue. For example, we want to see the whole screen structure. Is the screen structure ruptured or not? Okay, we want to see the whole lung structure. Is the lung injured during when you take the drug or not? So these are what the scanning objective lens can help. So this is when you will use it to scan the gross structure of the tissue. Then second one is 10x, yellow color. 10x, this one is the so-called low power objective lens. Low power objective lens is normally when you want to see the structure of the cell. You want to see the whole cell. You want to see the whole cell, how the structure of the whole cell, the shape of the cell, whether it actually cremated or not, something like that. Okay, and this one we will use the low power objective lens. Okay, then next blue color 40x. 40x blue color. This one is high power objective lens. High power. Okay, high power objective lens. This one is normally we will use it to zoom in. Okay, to see the detail. To see the detail in the structure. So, for example, you want to see the vacuole. For example, you want to see the cell wall. You want to see what is inside, not what is inside. Inside cannot see. Okay, you want to see the shape of the nucleus. This one, yes.
or when the cell is too small, then yes, you can use the high power. Okay. Then lastly, 100x. 100x means immersion oil, or sometimes it's called oil immersion. Immersion oil, oil immersion objective is the same. Okay. So this one, when we are going to use it, we were going to use it to zoom in for the very detailed structure. Okay, very detailed structure. Okay, and for those structure that's very small, for example, bacteria, we will use the immersion oil objective lens. Okay, so this is how we choose the objective lens. Okay, all right. Now for the 100x objective lens, it is called immersion oil. This one is called immersion oil objective lens. Why? Because when you use this objective lens, right, you must add the immersion oil. These three you cannot add. This one you must add. Okay? This one you need to remember. Yeah? These three you never ever touch the immersion oil. Yeah? You touch the immersion oil, you will spoil. Yeah? Then you're touching again. Okay? This one you must add the immersion oil. If you don't add the immersion oil, touching again, same thing. Yeah? So remember this one. Yeah? In practical 2, you are going to play with the microscope that's around like 5,000 to 6,000. Yes, it's not the normal microscope that you use in the school. Ours one is Leica, Olympus, it's all the branded version. And that's the one that is really used in the medical lab. Okay? We don't have. Electron microscope only government hospitals don't have. Only government university got it. Only a few research university got it. UKM, UPM. Last time in UPM I used that before. So it's very hard to use. Okay. Okay. So this one immersion oil, we won't let you to touch the electron microscope. Even if we have it, also we won't let you touch it. You know how much it costs for one electron microscope? If let's say it's TEM, it's around 2M. Yes. So you imagine a spreadsheet 2M, are you going to pay? Okay. SEM is even worse. SEM 3.5 to 4M. Yes. Okay, so if let's say you have the pocket money, you can ask your parents to buy one for you and play at home. Okay. So you don't have that. That is too expensive. Okay. So this one, immersion oil. Back to this one. Immersion oil, objective lens. Okay. You must always add the immersion oil before you can use it. Okay. Others cannot. Others you cannot touch. Okay. Okay. So why? Well, it's all due to the light. This one, well, there will be always the distance of the lens and your specimen, your glass slide. Okay. So this one, the distance is called the so-called working distance. It's called the working distance. Working distance. So, well, for 4x, 10x, 40x, the working distance is still okay. Okay? But 100x is so close. It is very, very close. Okay? You will feel it during the practical too. Yeah, you will see it by yourself. Okay? So, now, because of working distance and also the size of the lens, okay? 4x, 10x, 40x, after the light come out from the glass, which is higher density, air, which is lower density, right? So you have learned in the physics, from high density into low density, refraction occurs. Light refraction occurs. Okay? So refraction will occur and then it will bend the light. So for the 4x, 10x, 40x, even though the light refracted, but then it will still be captured. All of it will still be captured by the lens. So that's why no problem. They don't need the immersion oil. But then for 100x, for 100x, if let's say you don't put the immersion oil, what will happen is that they will refract. So when they refract, the lens is too small already. Some of the light will not be captured. When it cannot be captured, right? So that means that some of the light will be lost. So when it lost, only some of the light will be captured by the lens. So the image that comes into your eye will be blurred. Yeah, it will be blurred. So that's why if you don't put the immersion oil, image will be totally blurred. You can't see anything. Okay, so that's why you need to add the immersion oil. When you add the immersion oil, the immersion oil, the density is almost the same.
So that's why when the light, right, coming from the grass, okay, going into immersion oil, it will prevent the bending. It will reduce the bending of the light. In other words, reduce the refraction of the light. So the light not much refracted, so most of the light, it will be still be captured into the lens, and then with that, it can create a clear image. So that's why you can see it. So that's why immersion oil, it must be added when you are using 100x objective lens. Okay. A levels, they will explain in different way. A level, they don't explain in this way. A level, they will say that, okay, the immersion oil is having high refractive index. Okay, high refractive index. So that's why the light don't bend that much. So with that, they will increase the numerical aperture. Meaning that numerical aperture means that the ability of the lens to gather the light and resolving the specimen. Okay, so that's the numerical aperture. So that's their keyword in their marking scheme actually. Okay, but for us, not really. For us, if you want to explain in this way also can. You want to explain in this way also can. Either one. Yeah, I will accept both. Yeah, so don't worry. All the markers will accept both, so don't worry on that. Okay. All right, then next will be the calculation. Okay. Calculation before we go further. All right. First thing first is the conversion of the unit. Yeah, conversion of the unit. Okay. So for this one, I think you might think that it looks very simple. Okay, but this is one of the major careless mistake that a lot of students did. Okay, and there's a lot of the students that actually lost the whole 3 marks in the SAQ. Okay, because in SAQ we are normally SAQ for calculation 2 marks to 3 marks. Okay, and in bio there won't be any error carry forward. That it will not be error carry forward. Okay, so for example, the first step you need to calculate the magnification first. Okay, but when you calculate, you need to convert the unit. When you convert the unit, cm, convert to micrometer, you accidentally times 100,000. Alright, then it will be wrong. So your magnification of your first step that you count is wrong. Then you apply to the second step and third step, of course, all the answer is wrong. Okay, in that case, you will get 0. You will get totally 0. There won't be any error carry forward. Normally for chemistry and biology, there won't be any carry forward. This is not maths. Maths there will be, but then chemistry and bio normally no. Okay, because you are in medical field, you are not in engineering field. In engineering field, you fail. I mean, you calculate it wrongly, careless mistake in the first step, then eventually you get the wrong answer. You can still save it. But imagine one day you work as pharmacist, you work as the specialist, right? You calculate the anesthetic, the LA, local anesthetic that you want to inject in the patient. If you first step you count wrongly, the final concentration of the drugs is wrong. You inject, for sure die. Someone is going to pay their life. Okay, this happened, yeah. I mean, that one I think should be. Who's that? One of our prime minister son-in-law. He go to just for tooth extraction, and then the doctor calculated wrongly. Just one minor mistake, so the final concentration is wrong. After inject, die. Can you imagine that? It's just like to extract the tooth, only die on the spot, right? So that's why, because of this, our requirement is very, very precise and specific. Okay, there is no error carry forward because you are dealing with people's life. One careless mistake means that somebody is going to pay the life. So that's why we need to have this habit. Okay, so there's no error carry forward. Okay, so because of this, okay, for those who are always making careless mistake, right? Okay, and this table should be able to help you. Okay, so once you go into the example, okay, when the exam start, immediately draw this down in the draft paper first. Okay, right? So what happens is that, okay, when you want to convert from bigger unit to become smaller unit, you need to times 1,000. Okay, for example, to convert from 1 mm to become micrometer, you need to times 1,000, so it becomes 1,000 micrometer. If you want to convert 1 mm to become nanometer, means you need to times 1,000 and times another 1,000. So it becomes 100,000 nanometer. Okay, right? So when you want to convert from smaller unit to become bigger unit, then you will divide by 1,000. Okay, so be always careful on this. Don't make this careless mistake. This is not worth it at all. Okay, in previous cohort, 125, a lot of the students actually know how to do the calculation, but because of the careless mistake, you can imagine 30% of them did this careless mistake. And that question is 3 marks. All 3 marks gone. Because of they convert. You know why? Because the question gives cm. The question gives 2 cm. Ask them to convert to micrometer. Now you know what happened? They times 1,000 and times another 1,000. They make this kind of mistake. cm convert to mm is just times 10 only, but 30% of the students, they actually times 1,000 here. So total thing, the whole answer gone, 3 marks. The whole paper is 30 marks. 3 marks out of 30 is 10%. 10% is too great. So from A you drop to B plus. From B plus you drop to B minus A. So just because of this careless mistake. Okay. So please remember. Okay. Don't make this careless mistake. Okay. All right. So if let's say you always make careless mistake one, quickly draw this in the draft paper. Okay. And then during the exam, when the exam start, only you draw. Before exam start, you draw is considered as a plagiarism already. Okay, so after the exam start, only you draw it out. Okay, then any calculation you refer back to this. Then you make sure you don't make the careless mistake. Okay, not worth it. Yeah. Okay. So done this one. Okay, so before that, we quickly go through the definition. Okay, so 3 things. Contrast as the first thing. Contrast is actually the difference in the intensity from the specimen and the background, the object and the background. How well you can differentiate the object from the background, that is the contrast. Okay, that's the definition. So to increase the contrast, there are 2 ways. First way in microscopy, 2 ways. First way is you stain it up. You do the staining, then you will increase the contrast. Or if not, you will use the phase contrast. That means that you use the light that is in phase and out of phase to increase the contrast. That means use the phase contrast microscope. Phase contrast microscope, you can see that. Okay, you can see over here, you don't see any cilia. But then by using a phase contrast microscope, you can see, oh, actually they have the cilia here. Okay, so increase the contrast. Then that means that you can differentiate it from the background pattern. Okay. The second one is magnification. Magnification means that how large the image is being enlarged compared to their actual size. That's magnification. So for this one, we will always apply the formula. Magnification equals to the size of the image divided by the size of the actual. Okay, so it's MIA. Okay, MIA. So we will apply the magnification formula later. In solving the question. Okay. Then lastly is resolution. Resolution means that the ability to distinguish 2 points as 2 separate points. Okay. In other words, is that how detailed you can see. How detailed you can see. The higher the resolution, the higher resolving power means that the more detail you can see. Okay. So what it means by ability to distinguish 2 points as 2 points, how is it going to relate to this? Okay, very simple. Now I draw the 2 points here. I think easily you can differentiate this is 2 points. Okay, you can easily differentiate it. Okay, but if let's say I draw it like this, I think some of you at the back you will see this as 1 point. Some of you who are sitting very close, you will see this as 2 points. Why? Because the distance in between the 2 points is too short already. Okay, so when it's too short, right, you won't be able to separate the 2 points. When you're not able to separate the 2 points,
to you, the image that produced in your eye, it will be blurred. So that's why you will thought that it's only 1 point. Okay? So when this applies to our bare eye, why we cannot see the red blood cell? When you accidentally cut yourself, there will be bleeding, blood coming out. But you only see red. You won't be able to see the red blood cell one by one coming out. You can't. Why? It's right in front of you, but you cannot see it. Why? Because the resolution, the resolving power of your eye is not high enough. Okay? For example, your eye, what is the resolving power of your eye? Of your bare eye? No idea, it's your eye. Look into your manual. How many, every product got one manual, check your manual. What is the resolving power? All the eye, in general, is 100 micron. That means 0.1 mm. Anything smaller than 0.1 mm to you is blurred, you cannot see it. Even though it's in front of you, for example, bacteria right now is in front of me, I cannot see it. Because to me, it's blurred. Okay? Right, so this is our eye, but some of the people, they will have super eye. Super eye, they can observe until 80 to 90 micron. Smaller, they can see smaller things. Okay? So, how about the red blood cell? Why you cannot see the red blood cell? Because red blood cell, the size is around like this. Red blood cell is around 40 micron, the diameter. Okay? So why you cannot see it? You cannot see the red blood cell. Because the point here and the point here is only 40 microns. The distance is smaller than your resolving power. The distance is smaller than your resolution. So you cannot differentiate these 2 points as 2 points. You cannot differentiate this plus 1 and red and this plus 1 and red. So to you, the image that's being produced is red blur. That's it. If let's say you have a resolution that is smaller than 40, then what will happen? You will be able to differentiate these 2 points as 2 points. When you're able to differentiate these 2 points as 2 distinguished points, then that will mean that you will create a clear image here. So you can see the red blood cell rolling one by one. Okay, but too bad our bare eye is cannot. Okay, that's why we need microscope. Okay, so that is the meaning of resolution. Okay, resolution is actually very related to wavelength. In microscopy, it's very related to the wavelength. Okay, so what's the relationship? So this one, yeah, you need to know the concept. When you increase the wavelength, right, it is going to reduce the resolution or reduce the resolving power. Okay, so longer wavelength, lower resolution. Lower resolution means less detail you can see. Less detail you can see. Okay, and on the other hand, when you decrease the wavelength, then you will increase the resolution, and it will mean that you can see more detail. That is the relationship that you need to know about the resolution and the wavelength. Okay, so done with this, then now we can move on to the calculation. Okay, so for calculation, we look at the first part first, yeah? Okay, we look at the first part first. Okay, first part, first category, this is the question where they will want you to apply the magnification formula. For this one, normally the question will give you either the magnification or they will give you the measurement. Okay, so don't confuse that. Every time you have to figure out what type of question is this first. Okay, so let's look at the first one, straightforward one. Straightforward is that the question directly gives you the measurement. The question directly gives you the number. They tell you image size is 30 mm, and then the object size, actual size is 3 micrometer. Ask you to calculate the magnification. This is straightforward, very simple. So how to do this, how to solve this, you can just apply the formula. Magnification equals to the image over the actual. Okay. Then what is the first step? Substitute the number into the formula. Okay. So magnification is what we want to find. Image size is 30 mm. Okay.
actual size is the object, yeah? So that means it's 3 micrometer. And this is where a lot of students will make mistake, convert to the same unit. Okay? So mm you need to convert to the same unit first. Only then you can do the calculation. Okay? So you cannot do the calculation first, then only you convert. Cannot. You need to convert first. Convert the unit first. So mm convert to micrometer because most of the time the cell size is in micrometer. Okay, so that's why in order to convert the mm to become micrometer, I will times 1000 because mm to become micrometer is 1000, yeah, times 1000. So magnification will become 30,000 micrometer divided by 3 micrometer. So magnification equals to 10,000 times magnification. Okay, micrometer slash. All right, so with that, that is the final answer, magnification. This is the first type of question, straightforward question. Okay. Then next, second type of question. Second type of question is that we will give you an image. And we will give you an image. We won't tell you what is the measurement. Okay, we will give you an image and then you have to measure yourself. Okay, but now it is using OAS plus BYOD. BYOD different computer. Oh, you are using your own computer. So different computer, different screen size. Okay, so that's why you cannot use your ruler to measure on your screen. Because you might enlarge, you might zoom in and go and measure not enough. Okay, so there will be normally having a virtual ruler like this. Or we will directly show you what's the measurement. Okay, so if we show you the measurement, you use the measurement. If let's say we don't show you the measurement, there should be a virtual ruler. This virtual ruler, it will be same size with the image. So that means it will be fixed with the image. If you zoom in the image, the virtual ruler will zoom in also. Okay, so that's why you have to always use the virtual ruler if it's given. Last again, I don't want to tell you who is it. Some students they actually use their ruler to measure on their screen. And of course, they get wrong, of course. Okay? And then got one super lucky student, because she zoomed out, then the size of the cell becomes smaller, and then he used the ruler to measure on the screen. And then the measurement is exactly the same like the so lucky. He is so lucky, but only one. I don't think you will be that lucky. Okay? So don't do that. If virtual ruler given means you must use the virtual ruler. Okay? Alright, so now this is the second type of the question, image given. If image given, what they want to find? They want to find the size of the organelle. Means that they want to find the actual size. Okay, they want to find the actual size. They tell you magnification is 100,000. Okay, so how to do this? Same thing. Magnification equals to the image over the actual. What is the first step to do? First step is you need to measure. You need to make the measurement. That's the first step to do. Okay, so that's why in the mind map you can see, you need to make the measurement of the image first. Get the measurement of the image. Okay. So let's say virtual ruler given here. So you have to measure. Okay. So let's say it is from 1 cm until, let's say 9.5 cm. Okay. Check the ruler also. Sometimes ruler given is cm, sometimes mm. Okay. So this one, let's say, is cm. All right. So what is the magnification? Magnification has been given here. Is 100,000. What is the image size? Image size is 9.5 cm, right? So 9.5 cm, normally I don't like to write cm. I directly change to mm already. Then only I write it down. What happened is that a lot of students they write 9.5 cm and then when they convert later they have problems. The 30% of the students. So many of them. Okay, so I will directly write 95 mm. I don't want to use cm. I use mm. No wrong, nothing wrong. It's okay. 95 mm. Okay, divide by the actual is what we want to find. Okay, so I will rearrange the equation. It becomes a over 95 mm. I want to convert to. I will micrometer first. Normally for me, I will convert first because if you convert at last, sometimes it will make a mistake also. Okay, so normally I will convert directly. So 95 mm, I want to change to micrometer, I will times 1000. So it becomes 95,000 micrometer divided by the 100,000. Okay, so with that, what is the actual size? Actual size is 0.95 micrometer. So that is the final answer. So that's the actual size. Yep. Oh yeah, it should be 8.5. Sorry. I see careless mistake again. Gone. This one, 2 marks gone. 85. So this one all 2 marks gone. So don't make mistake like me. Okay. Okay, because it's from 1 until 9.5, so it's total 8.5 cm only. Yeah, 8.5 cm. So 0.85 micrometer. Okay. Done, yeah? Then next, third type of question. Third type of question is scale bar given. Scale bar given means that they will give you an image, okay, and then they tell you nothing. No magnification, no size, no other thing. They only give you one scale bar like this. They will give you a scale bar like this. Okay, so they will tell you, okay, this scale bar is 2 micrometer. Is it 2 micrometer? Is it that long, 2 micrometer? No, of course not. Okay, so that means that this 2 micrometer is actually the actual size. Okay, the actual size. So you make the measurement for this scale bar, you will know the image size of the scale bar. Then when you have the image size, you have the actual size, you can know what is the magnification. Okay, so that's why the first step for this third type of the question, third type of question is scale bar given. Scale bar given type question, the first step that you need to do is use the scale bar to find the magnification of this picture first. Because scale bar and the picture, they come together. So that's why this scale bar and this picture, they are actually of the same magnification. Since for the picture, the image, they don't give us anything, so we don't know. We got no clue at all. So that's why we need to use the scale bar to find the magnification first. So that's our first step. Okay? So how do we use the scale bar to find the magnification? So the first step, same thing, we will apply the magnification formula. Okay? So magnification is what we want to find. Image size is what we can actually measure. Okay? Because it's the same thing. For example, I have one cell like this. I tell you this whole thing is 2 micron. The diameter of the cell is 2 micron. Is it 2 micron like this? So big? Cannot. So there must be an image size also, right? So can you find the magnification of this cell? Of course, why not? Because you can measure the length, you get the image size. You divide by the actual size, you get the magnification. So same thing, same concept applies to this scale bar as well. So for this scale bar, well, magnification is what I want to find. Image size is what I need to measure. Okay, same thing. They will normally give you a virtual ruler. Okay, so the virtual ruler, let's say 2.5, let's say. Okay, 2.5. We make it simple. Let's say 2.5. Let's say here to here is 2.5 cm, let's say. Okay, so 2.5 cm, that means that it is 25 mm. Divide by the actual size. What is the actual size? Actual size is 2 micrometer. So it's 2 micrometer. So you need to convert to the same unit first. Micrometer and millimeter, you cannot directly divide. Yeah? So you have to convert. So magnification equals to 25 mm times 1000. It becomes 25,000 micrometer. So now it's same unit. Okay. So what is the magnification? Magnification will be 12500 magnification. Okay, so once you have the magnification, it means that you know that what is the magnification of this image. You know the magnification of the image. You also know the image size because you can measure it. Can you find the actual size? Why not? Okay, So that's why, in order to find the size of this organelle, so we can apply the formula again. And we can apply the formula again. Magnification, we have just calculated that. It's 12500. Okay. Image size, you have to measure. Image size, you measure from here. 1. Let's say until 9.5, say 10.5, right? So it's 9.5 cm. No? Okay, the measurement is 9.5 cm from here to here. We use the virtual ruler. Okay, so it is 9.5 cm means 95 mm. Okay, 95 mm. Yeah. Actual size is the one we want to find. Okay, so with that, we rearrange the equation. Actual size equals to 95 mm convert to micrometer times 1000, that is micrometer, divide by 12500. Okay, so what is the actual size? The calculator. Calculator, anyone with calculator? 7.6. 7.6 micrometer. So that means that this chloroplast is 7.6 micrometer. Okay, so that is the calculation. Okay, so with that we have done with the 3 types of the question here. Okay, so for the last part is the measuring cell. Yeah, measuring cell. This one I think we will move it to the next class, in the next tutorial. But this one is quite simple, so I think I will try to finish it now. Okay, the measuring estimation via the diameter. All right. So quickly we go through that. Yeah. Okay. Measuring of the cell. Yeah. Measuring of the cell. That type of question it will involve eyepiece strategy and stage micrometer. Eyepiece and stage micrometer. No matter what type of this, it is always under the microscope. Yeah. It will be always giving you 100 division. It will be a line with 100 division. Okay, so 1 division, either you can call it as 1 division, or you can call it as 1 pitch. Pitch, P-I-T-C-H, 1 pitch. Okay, so under the microscope, yeah, it's always 100. Yeah? 1 line with 100 division or 100 pitch. No matter for this one or this one, it's the same. Okay, so that's the first thing that you need to know. Second thing, for eyepiece strategy, 1 pitch, normally I will write it as 1 PE. 1 pitch on the eyepiece strategy, 1 PE. While stage micrometer, I write 1 PS, 1 pitch on the stage micrometer. You can use this short form in the calculation. Because if not, then you have to write 1 division on the eyepiece strategy equals to what? You have to write a lot. Okay? So it is better you can use the short form, 1 PE or 1 PS to differentiate it. Okay? Right. So this one, no problem. That's it. This one we will talk about this first because we are going to use this for the estimation. Okay. For the questions related to this. For the question related to this, right? In the question, you will only see stage micrometer given. No other thing. No eyepiece reticule. No other thing given. Only stage micrometer given. If only stage micrometer given is this one. Okay, then you have to follow the step. Okay, so before that, for stage micrometer, you need to know that there will be two types of the stage micrometer. We call it 2 scale. So one type is called 1 mm scale. The question will tell you the stage micrometer with the scale of 1 mm. Another one is called with the scale of 1 cm. This is important to complete our first step because our first step is to find 1 PS. Okay, you can see here, our first step is to find 1 PS. Yeah? Okay, so scale is important. In the question, they will state that. Okay, so if let's say the question is the scale of 1 mm, so that means that it always means that 100 PS equals to 1 mm. So 100 PS is equals to 1000 micrometer. 1 PS is equals to 1000 micrometer divided by 100. 1 PS is equals to 10 micron. This is fixed. This is confirmed. Even though in the question sometimes you don't see 100 division, 100 pitch of the stage micrometer also, as long as they tell you that the stage micrometer is with the scale of 1 mm, that means that 1 PS is 10 micron. This is confirmed. Okay? If let's say it's 1 cm scale, 1 cm scale will mean that 100 PS is always equals to 1 cm. And that will mean that 1 PS will be equals to 100 micron. That is fixed also.
You can try the calculation like this.
So this is the thing that you need to remember.
1 mm scale means 1 PS equals to 10 micron. It's always how to show the calculation is always 100 PS equals to 1 mm. 1 cm scale is always 100 PS equals to 1 cm. This is a must. Even though in the image is given, the stage micrometer only got 30 division or so, we don't care. To find the 1 PS, we follow the scale only. Okay?
So once you understand this, then now we can see the questions. So how the first question type will be?
Okay, first question type will be is that they will tell you that, okay, they will show you a stage micrometer to measure the diameter of this microscopic view.
Okay, so to solve this question, we call it estimation of the cell size. Estimation of the cell size, 3 steps. Yeah. So first step is find 1 PS. Okay,
so from the question, what is the scale of the stage micrometer? The scale of stage micrometer is 1 mm.
So that's why you need to show the equation. Yeah, 100 PS equals to 1 mm. So with that, 1 PS equals to 1000 micron divided by 100. So 1 PS equals to 10 micron. That is the first step. So that means what? That means that this one pitch on the stage micrometer is equals to 10 micron. That's what it means.
Why you want to do the first one?
So this is step number 1. Okay. Then step number 2. Step number 2 is you need to find the diameter of the microscopic view.
You need to find the diameter of microscopic view. So the microscopic view got how many PS? How many division? From 0 until 30, right?
That's the diameter, right?
So the diameter is equal to 30 PS, right?
Okay. So diameter is equals to 30 PS.
Because got 30, right? You count that's the diameter, across the diameter. Okay.
So just now we count 1 PS is 10 micron, right?
So 30 PS equals to?
So you times 30 times 10 micron, you get 300 micron. So that means that this is the diameter. Diameter is 300 micron. Okay? And then last step is estimate the size of the cell using the diameter.
So normally the question will tell you across the diameter of the microscopic view got how many cells? Over here they tell you that across the diameter of the microscopic view got 1 cell, 2 cells. So estimate the size of this cell. Can you do that? Of course. So diameter divided by
number of the cells. So to estimate it, size of the cell equals to diameter of the microscopic view divided by number of the cell. Okay, so the diameter just now we calculate is 300 micron. Number of the cell 1 and 2. So that's why it is 150 micrometer. So that's how you estimate the size of the cell.
If they say they tell you that across the microscopic view, across the diameter of the microscopic view got 5 cells, then you divide by 5. Okay, so that's the 3 steps for this
first type of question, estimation.
Okay, right. So this one actually is quite simple. Yeah, it's actually quite simple. The only thing is here, you need to find the 1 PS. If you can find the 1 PS, the rest actually logically you can solve it. Okay, so this one is easy question. Another type, that one is harder. Okay, that one I will need to use some time. So we will discuss on this Friday. Okay, so on this Friday we will complete the tutorial, right? So try you do the tutorial 1 and 2, yeah, both tutorials try to do. Then we try to finish. Okay, you cannot finish, then we just move to the next class, right? So it's fine. Okay, so that's why do both the tutorial. Yeah, do both the tutorial. Okay.
So with that, I think this basically all. Right. So thank you for your lunch time. Okay, so I think we go for lunch. Okay. So thank you and bye bye.
I want to ask, how do you type micrometer on the laptop?
Oh, you can use U.
Use your.
Use UMM.
Okay.
Yeah. Yeah.